Hi, everyone. My name is Yingjie, and my research is kind of similar to Yuxian's. The difference is that she look at air pollution, which is the pollution above the land. And my research is about pollution below the water. Sorry. Yeah, so as you can see from this background, there are several hotspots there. Actually, research have found that across the globe, there are more than 500 dead zones in the coastal area. So this number is still increasing because like 10 years ago, there is a review paper published in Science. And in that review, they identify synthesis like 400 dead zones. But 10 years later, they are increased to more than 500. So similar here, you can see the red dot, each red dot represents a dead zone identified by some literatures. So one key question is that why should we care? As a scientist, also focused on environmental science, I do care because we would like to protect the environment and conserve the biodiversity in the coastal zones. I guess some of you also care because you are in the ESPP program. And more importantly, as you can see, the dead zones can cause fishery degradation. So across globe, there are 3 billion people are relying on the coastal zones for their livelihood. So if all the coastal zones are degraded and people may lose their livelihood. And one close example is that in our Gulf of Mexico area, the dead zone, the area of dead zone is increasingly repeatedly every year. So in 2017, the total area of dead zone in our Gulf is almost equal to the area of New Jersey. So there is a joke that the senator of New Jersey was complaining that can we do not use New Jersey? I think the point there is that can we reduce the size of the dead zones and do not play jokes with the state of New Jersey. And also another chapter also looking at the Gulf of Mexico area, and previously the Gulf of Mexico provided 85% of the shrimp, 60% of oyster, and 50% of recreational fishing, and 30% of the blue crab for the US. But in the most recent decades, the US is importing more and more seafood from East Asia, like China and Indonesia. So it seems the degradation of our Gulf is causing some problems, especially because of the dead zones. So there is intensive research looking at the problem and identify that it's most caused by the nutrient flow from the Middle West. But one key question is that how the dead zones change over time and space. Currently, there are several studies, but that do some limitations. For example, there was one very famous marine scientist in Louisiana State University, Professor Nancy Rabelais. And so each year since 1985, so each year they dropped crews and went to the Gulf area. As you can see, they were sampling the seawater, and test the dissolved oxygen level across the whole region. And then they report a map like the right-hand corner and calculate and they report a total area as I mentioned earlier, the size in 2017 is almost equal to New Jersey. Okay. So same here. The area also generally increase over years. But there are some limitations that, for example, in 2016, they were not able to take water samplings because of funding issues and some other issues. And especially for this year, because of global pandemic, that may another potential disturbance, they may not able to take the water samplings. And besides, there are several other limitations. For example, this kind of research, the field observation research, is kind of like a labor intensive and time consuming and also costly. So when I was taking Dr. Chi's remote sensing class, I began to thinking whether we can combine the remote sensing data with the observational data to map or monitoring the change in the Gulf of Mexico area. So on one hand, we have tons of data. As you can see, a bunch of agencies are collecting water sampling in the Gulf. And also cover a long time period, and the spatial coverage is also pretty good. So in total, since 1985, there are like around 8,000 sampling locations, and also we have I have able to collect the sampling data point from all these agencies and got around 800,000 data points. So that's that in that data record we have the specific date and the time and what's the oxygen dissolved oxygen level and at what and a sample at water level below the water, and also associated with other related information. So specifically, I highlight two data sources, like the CMAP, which is a Southeast Area Monitoring and Assessment Program, and also the LUMCON, which is Louisiana University's Marine Consortium. So these two agencies kind of like, they have most of the data and have pretty good coverage. And on another hand, we have pretty good remote sensing data images. For example, the MODIS have ocean colors and at a daily level and across the globe. So also there are several other data images. For example, they available. For example, when surface sea surface temperature, precipitation, and solar irradiance, and several other like water density, such kind of maps. So so I was wondering, could we just synthesize two kind of data set? One is the observational data, and another is a bunch of remote sensing data, and input into machine learning algorithm, and boom, we can produce such a map and to monitoring the data zone change across the globe. And also there is another very powerful free cloud-based computing platform. It's Google Earth Engine, which I also learned from Dr. Chi's class. So all this seems pretty promising to generate very fantastic result. But is that so easy? When I really start the project, I kind of like let the reality punch on my face. So there are so many difficulties. So first, there are very complicated process. First, the nitrogen flow and down to the Gulf of Mexico area, and it takes time. And then because the nutrient-rich water flow in, the algae begins to grow. And then it takes several days and they die and sink down to the water, and bacteria deplete the water to I don't remember the words, but the bacteria deplete the water of oxygen to let's see, to consume the algae. So all along this process, because of the consumption of oxygen below the water, that can cause the dead zones. But imagine that if you hold a glass of water, and this glass of water has low oxygen, even at this very close distance, I guess we cannot identify whether it's low oxygen or high oxygen level. So one possible way is that can we identify the linkage between something we can observe from the remote sensing data from the space, and then such a thing can relate to the low oxygen level below the water. So I play with the data. As you can see here, the red bar indicate here, figure 8, indicate the low dissolved oxygen was detected at this date, for example, roughly June 22. And I plotted the chlorophyll A, which is taken from the remote sensing images, and we can identify roughly several weeks ago, there is a peak in chlorophyll A. So this seems some linkages as we imagine the algae bloomed and then take several weeks, grow and die, and then sink down to the water and cause dead zone. So, but the first thing is that we need to identify what's the time lag. After several rounds of the data exploration, I identify like a 1 week to 3 weeks is there is a good correlation between the reflectance of the remote sensing image at different band. So each plot at each plot it means a different band and versus the dissolved oxygen level. So, but over 4 weeks before, there are pretty low correlation. And this figure may show a better sense. For example, the left-hand figure A shows in the locations where dead zones were identified. You may notice there is pretty high chlorophyll A, but for other locations, sampling locations, there are no dead zones. The chlorophyll A is pretty low. And further, we may want to know, before we input all data into the model, we need to address some like a collinearity issues. Even we're using random forest or other machine learning algorithm, it's in nature they are regression or correlation analysis. So we may need to identify which remote sensing band are not good for the model. So in this figure, we may find that chlorophyll A and POC and several highlighted remote sensing band. Here, the red box means oxygen level below 2 milligram per liter, which can be an indicator of that zone. So for example, chlorophyll A is pretty, so here is like a significant test. So if the label here, A, C, B, A, B, so if the letters here are different, which means these two groups are significantly different from each other. So yeah, here you can see we can identify this band. So after that, we input the data into the model and training the model, and did a take 2017 example and generated this map. And after we compare with the Louisiana Professor Nancy Rablas group as their result, you can see some pretty good overlap, but you can also find our method can pretty well detect area. For example, here, they didn't have any sampling point there. But further, at a current stage, this research is still at a preliminary analysis stage. So for the further steps, I think the first one, we need to compare with other people's research. They are using biological models or geospatial models to simulate and generate the data zone maps. So our method can compare with them and then tune our model. And secondly, I plan to apply our model to larger scale. For example, the UNEP and Intergovernmental Oceanographic Commission identified 63 coastal zones which are at high risk of pollution. So we may first choose several of them and apply our algorithm to such area and do a mapping. So why is it significant? Because life below water is one of the 17 Sustainable Development Goals proposed by the United Nations. And second, our method is kind of replicable, reproducible, and can be applied to other areas to help with the global coastal assessment. And our result, we may have potential to inform stakeholders to develop integrated strategies for marine conservation. And finally, I would thank several other collaborators who provide data and help, and also the ESPP Summer Fellowship, the great support. Thank you all.
Thank you. We have time for one quick question. If anyone would like to put that in the chat or raise your hand.